Hello everyone, in this video I'll talk about the ultrastructure of an animal and then a plant cell. Well, the ultrastructure means the fine detailed structure that can be revealed by the electron microscope. And if you remember, at IGCSE level, candidates are supposed to know only the structure of the cells that can be revealed by light microscope, which does not provide fine detail. But as you can see in this micrograph here, the cell is not actually as empty as we've drawn it and learned about it in the IGCSE level. There are many other structures found in the cell. So eukaryotic cells have tiny compartments with unique particular functions that they carry out. So there is definitely a division of labor within the cell. So cell is like a miniature city. There's a center of control. There's a power plant. There are internal transportation units. There's a food factory, the disposal unit. So like a miniature city, there's the division of labor going on within the cell. Okay, let's begin with the nucleus. Nucleus is the largest cell organelle, and it is surrounded by a double membrane known as the nuclear envelope. The nuclear envelope is perforated by nuclear pores, which are actually associations of proteins that provide exchange between the cytoplasm and the nucleus. Nucleus contains the genetic material, the DNA. And within the nucleus, the DNA is organized into discrete units called the chromosome. Like you can see here, the chromosomes become visible during cell division, which we'll see in another chapter. Unlike the prokaryotic DNA, the bacterial DNA, the eukaryotic DNA is wrapped around globular proteins named as histone proteins. So the complex of DNA wrapped around histone proteins is called as the chromatin. Well, even though the discrete chromosomes are present in a cell, but if the cell is not dividing, the stained chromatin appears as a different mass in micrographs, and the chromosomes cannot be distinguished from one another. But however, as a cell prepares to divide, the chromosomes coil, we say condense further, becoming thick enough to be distinguished as separate structures, as you can see here in this micrograph. So the chromosomes are visible now. In a nucleus of non-dividing cell, we can also see a mass of densely stained granules and the fibers adjoining part of the chromatin. So here, this dense area, as you can see, called as a nucleolus. A type of RNA that we'll see in another chapter called the ribosomal RNA or rRNA is synthesized from the instructions in the DNA in nucleolus. Also again in the nucleolus, proteins imported from the cytoplasm are assembled with rRNA into large and small subunits of ribosome. I need to talk about a technical detail of the nuclear envelope here, and here's a past paper question. Which pair of organelles has internal membranes? Well, it may seem a bit early to answer this question since we didn't know any of these organelles yet, but let me just explain for you. The chloroplasts, the mitochondria, also have double membrane, including the nucleus as well. So which one is the correct answer? Is it A or is it B? Chloroplast mitochondria or chloroplast nuclei? Well, here is the difference. A double membrane also surrounds the nucleus. However, the nuclear envelope is not continuous, but instead consists of a set of closed membranes that come together at the structures called the nuclear pores. Well, technically, the double membrane of the nuclear envelope is not considered as having an internal membrane. So what is the difference? Let me try to sketch. So that's the mitochondrion, the outer membrane, and here is the inner membrane. So both of them are continuous. Or a chloroplast, outer membrane, and a continuous inner membrane. But if we close up the nuclear envelope, the double membrane is like this. And here the nuclear envelope has the nuclear pores. So it's not continuous. Well, since there are two layers of membrane to pass through, we call it double membrane, but this membrane and this membrane are not considered as being either external or internal, since the membrane here is not continuous, as you can see here. Forms a closed membrane at the nuclear pore. So that's the technical difference. So the answer to this question should be A, not B. Ribosomes are the sites of protein synthesis, and ribosomes are not membrane-covered organelles. Ribosomes are very small complexes, only about 25 nanometers in diameter, which consist of rRNA, ribosomal RNA, and the small and the large subunit, protein subunit. They're common structures in all cells, but the prokaryotic cells, the bacterial cells, have a smaller size ribosome, 70S ribosome, compared to the eukaryotic ribosome, which is being ADS. So even in eukaryotic cells, organelles such as mitochondria and the chloroplast may also have 70S ribosome in them. Endoplasmic reticulum is an extensive system of membranes starting right around the nucleus, running through the cytoplasm. And the membranes form an extended system of flattened compartments called as the sacs. And these sacs may also be interconnected. There are two types of endoplasmic reticulum, rough and smooth. Rough endoplasmic reticulum, as the name implies, has tiny bumps, the organelles ribosomes on it. Ribosomes can be found freely in the cytoplasm or maybe attached on the endoplasmic reticulum, forming the rough one. Since rough endoplasmic reticulum has ribosomes, its main job is protein synthesis. And the smooth endoplasmic reticulum, having no ribosome, is involved in lipid synthesis. The proteins and the lipids formed in the endoplasmic reticulum then travel to the Golgi body or Golgi apparatus. Golgi serves like a warehouse for receiving, sorting, shipping, and even some manufacturing. So the final modification and packaging is done here. So the products of endoplasmic reticulum, such as the proteins, they are modified and stored and then sent to the other destinations. Golgi also consists of flattened membranous sacs. A Golgi sac has a distinct structural directionality, and it's easy to spot on a micrograph. It's like this. And there are two sides of the Golgi sac. So this is the cis face, the entry, and the exit is the trans phase. Okay, let's talk briefly about the endomembrane system, which includes the nuclear envelope, the endoplasmic reticulum, and the Golgi apparatus, lysosome, and maybe various kinds of vesicles and vacuoles, and even the plasma membrane, the cell surface membrane. Well, this system carries out a variety of tasks in the cell, including the synthesis of the protein, like in the rough endoplasmic reticulum, right here, so proteins are synthesized, or in the smooth endoplasmic reticulum, the lipids are synthesized, right? And then the proteins and the lipids need to be transported, right? So as the products of the endoplasmic reticulum leave out the endoplasmic reticulum, so they simply pinch off into a vesicle. So a vesicle is a tiny sac made of membrane. Later on, this vesicle then travels to the Golgi and literally bumps into the Golgi apparatus and pours its content into the Golgi apparatus. And then the product of the Golgi apparatus, as it leaves, again breaks off into a vesicle and then either travels internally inside the cell, right, or maybe out of the cell, will be excreted out. So not surprisingly, the Golgi apparatus is especially extensive in cells, which is specialized in secretion. Lysosomes are membranous sacs which consist of one single membrane only that contain hydrolytic enzymes or digestive enzymes that the animal cells use to digest the macromolecules. Lysosomes are responsible for the breakdown of unwanted structures such as the old organelles or even the whole cell, as in the mammary glands after lactation, for instance. In white blood cells, the lysosomes are used to digest the bacteria. We'll see very soon. The heads of the sperm contain a special lysosome, the acrosome we name it, for digesting the path to the ovum of the egg. Mitochondrion, or plural mitochondria, are site of aerobic respiration in eukaryotic cells. Mitochondria also consist of double membrane, smooth and continuous outer membrane, and inner membrane that makes foldings. So these foldings are to increase the surface area of the membrane, which basically holds enzymes for aerobic respiration. The inner membrane is called as a cristae, and the fluid is the matrix. So the fluid is the matrix. The energy released during the aerobic respiration reactions is stored in ATP molecules, so that's the currency of energy in the cell. So you have to be careful in how you write your answers in AS biology exams, since energy can neither be created nor be destroyed. We can't say energy is produced in mitochondria. What is in fact produced is the ATP molecules, or you can just say energy is released and ATP molecules are produced. Microtubules and MTOC, or microtubule organizing center. Well, microtubules are long, rigid, hollow tubes that are made up of tubulin proteins, alpha and beta tubulin protein here. So here you can see two tubulins forming a dimer and then a special arrangement which also leaves a hollow space and then forming a microtubule. So these microtubules are important elements of the cytoskeleton along with microfilaments and the intermediate filaments, which are actually not the topic of AS biology. Cytoskeleton supports the cell, gives the cell its shape, and the membrane-bound organelles are held in place in the cell by the cytoskeleton. The secretory vesicles, for instance, are moved within the cell along the surfaces of the microtubules. So microtubules also form an intracellular transport system, like a railway. Another function during the nuclear division, the spindle used for the separation of chromatids of the chromosomes is made of microtubules. And microtubules form part of the structure of the centriole as well. What's an MTOC? Well, MTOC is a special location in the cell which controls the microtubule formation from the tubulin proteins. A centriole is a hollow cylinder which actually consists of 9 triplets of microtubules, and which can be found in all animal cells and in most of the protists, but not in plant cells. Centrioles are also found at the base of the cilia and the flagella. Until recently, we used to think that centrioles form the spindle fibers during nuclear division, but in fact the function of the centrioles remain a mystery. It is now known that the spindle fibers are formed by the centrosome, not the centriole. And the centrosome is the region surrounding pairs of the centriole. Even the Cambridge was confused. Let me clear this out for you. Here is a question from 2015, a very recent question. Now check this out. In animal cells, centrioles are responsible for assembling microtubules to make the spindle, which is not accepted and not correct anymore. Right? So what do? We just replace the word centriole with centrosome from now on. As simple as it is. Chloroplasts are important organelles found in the plant cells. You'll be learning about the detailed structure and adaptation of the chloroplasts in A2 biology, but just for now, for the ultrastructure. The chloroplasts are also made up of double membrane, but unlike the mitochondria, both of the membranes are smooth, so it's like this, and the inner membrane also like that. Right? And in the inside of the chloroplast are stacks of membrane. And each stack consists of the disc-like structure called the thylakoid. So each stack of thylakoid disc called as the granum, right? And the fluid that fills in the chloroplast called the stroma. And very much like the mitochondria, chloroplasts also have their own DNA. Small circular DNA and 70S ribosome. A familiar organelle from IGCSE, the vacuole. Vacuole, remember, stores food, salts, pigments, and wastes. The difference between the animals and the plant cell. Plant cells have large and permanent vacuole. Well, some animal cells might have some temporary vacuoles as well. Right? Tonoplast is the membrane of the vacuole, and the content of the vacuole called as sap. The cell membrane of the plant cells is also surrounded by a rigid layer called the cell wall, which consists of the fibers of cellulose. The cell wall gives the plant a definite shape, and it prevents the cell from bursting when water enters by osmosis. Remember from IGCSE. The cell walls may also be reinforced with extracellulose, with a hard material called lignin, which makes the wood for extra strength. Cell walls are freely permeable or fully permeable, allowing free movements of molecules and ions through the cell surface membrane. The plant cells are linked to neighboring cells by means of fine strands of cytoplasm called as plasmodesma, or plasmodesmata in plural. So plasmodesmata are openings literally within the neighboring cells that allow the cytoplasm to stream within the neighboring cells. Here is a plant cell micrograph. Since the plant cells consist of large central permanent vacuole, the other organelles are like being pushed to the margins of the plant cell. And here are the chloroplasts that you can see. So what about these white area in the chloroplast? The starch grain starch grains. Chloroplasts are the sites of photosynthesis, and starch is the form of carbohydrate that is being stored. And here you can see it in the nucleus with dense nucleolus in it. So that's all for the ultrastructure of the animal cell and the plant cell. But for the exam, you need to know which structures can be seen only by the electron microscope or both with electron and light microscope, which will be in another video.